Um, Great. So with that, I guess we can get started. If anyone wants to, say anything or rather not type a question, and we'll give you time afterwards to speak on that. So I'm going to get started with our first presenter today. I have the honor of presenting Kyle Roy. Hi, Kyle. Kyle is a geneticist at the USGS Pacific Island Ecosystem Research Center, and she is also currently a PhD student at Purdue University. So she is currently in Indiana, calling in and going to give her presentation today on what we've learned from the 5 years of rapid ohia death entomology and our intentions for future ohia stewardship. So with that, take it away, Kyle.
Okay, awesome. I'm so happy to be here. I'm excited to meet a lot of you guys. I actually don't know you guys. So I tried to be a little bit reflective with this presentation and really talk about how we've been, what we've been doing for the past 5 years and really what we've learned and how we want to kind of shift gears of our own research. And so how it started back in 2015, rapid ohia death entomology was started by Dr. Gordon Bennett and Dr. Curtis Ewing. And I worked as a technician for them for a short amount of time. And then they moved on to other things, well, to California. And then the project shifted gears into and was taken over by US Geological Survey, where I work now, and also with Hawaii Cooperative Studies Unit. And so none of this work would be possible without our big team. And as you can see, that our team has really grown over the years as well. So we have Dr. Carter Atkinson, who he actually literally just retired last week, Thursday. So congratulations to him. Bob Peck, Ellen Dunkel, Kelly Janicki, Dan Micros, and Josie Tupu. And back when we started this research, we had no idea if insects or arthropods were even involved with rapid ohia death. We only had hypotheses based on other studies around the world. And 5 years later, we know that there are actually 5 different ambrosia beetle species that are involved with the spread of rapid ohia death. So what exactly are ambrosia beetles? So they're tiny little weevils, as you can see. They're about 3 to 5 millimeters. You can see how small they are on this piece of, on this dime. And so they live in the xylem of the trees, also where Ceratocystis lives. And so they have these special pockets called mycotangia that they keep their ambrosia fungi in, and they're actually fungus farmers. So they'll plant their ambrosia fungi that they use as their food. And what they do is they'll bore into a tree and create their home, which is called a gallery. And the beetles that we're dealing with in Hawaii are very invasive all over the world. They are in the tribe called Xylobarini, and they're known for being their ability to invade other areas because of they have a parthenogenic reproductive behavior where the males are haploid and the females are diploid. And so you could actually just have a single introduction of a female that could start an entire population in a new place. And so if their habitat is suitable, they can remain in that single gallery for many years. And these beetles are specifically attracted to ethanol. And this ethanol scent is emitted by stressed trees. So anything that is stressing a tree, such as invasive plant species, drought, nutrient limitation, flooding, fire, volcanoes, anything that will stress them can cause this ethanol reaction that will bring in the beetles. So we have found that there are 2 different ways that the ambrosia beetles are involved with the spread of rapid ohia death. And the first one is what we call indirect transmission. And so this refers to the movement of the frass that they create. And so their frass is actually these tiny little wood particles that are the same size as their mandibles. And so they're actually chewing the wood and kicking it back out into the environment. And when they're doing that, there could be ceratocystis spores or propagules embedded in the frass, and that's a way for the fungus to get from the inside of the tree to the outside environment. And this frass can then blow in the wind, fall into the soil and get moved in the soil, move on your boots that way, move in potted plants, and potentially even move in the water as well. And the other way that ambrosia beetles are involved with the spread is through direct transmission, and that's referring to the actual direct movement of spores on their bodies. And so ceratocystis produces these sexual fruiting structures called perithecia. And so at the tip, it puts out this ascospore mass, and it's sticky. And to show you guys just how sticky it is, you can see here, these are some beetles put in a culture, and you can see how sticky these ascospores are, and it's getting stuck in their bodies. So if they're leaving the whole gallery to go start a new family, they could get the spores stuck on their bodies and transfer the disease that way. So we have found 5 different ambrosia beetle species that can regularly reproduce in broad infected ohia. And what's interesting about these beetle species is that they're all invasive to Hawaii except for this one native, Xyloborus semilimus. And many of them are also associated with other diseases, and some of them other ceratocystis diseases as well. And so looking into the indirect transmission, so referring to the movement of frass, we have been able to find viable ceratocystis propagules in frass produced by all 5 of those ambrosia beetle species that I just mentioned earlier. And so all of them are capable of producing this frass and putting it out into the environment. The viability of the frass seems to depend on the stage of the infection of the tree. And you can find viable propagules can get kicked out into the environment from all different heights on the tree, the entire bowl. And so a way that we test for viability is through this carrot baiting technique. And so carrots are the best media for growing ceratocystis. And so you can see this is just a carrot disc, and there's frass here on top of it. And here you can see ceratocystis is this black mass growing out. And when this is normally in person, you guys usually come visit the lab. So I wanted to throw in some videos to make it a little bit more interactive. And so all the techs got together and made this awesome video to show you guys how carrot baiting works.
Hi everybody, I'm Ellen. I just wanted to show you around our lab down in Hilo a little bit, show you what we do when we are not in the field. One of the things we do is we identify beetles. So here's a bunch of beetles that we've caught in the field that I pinned.
Oh no!
And point mounted and identified and put nice pretty labels on them with the locality of where we caught them and the species. It doesn't look like it because they're so tiny, but when you look at them under the microscope, you can see there's like a bunch of different species. So that's a fun thing to do. And then over here we have our lab mates, Dan. He is looking through our fancy microscope to see if these carrot baits from samples we collected in the field are positive for ceratocystis. So he'll look at them under the microscope and look for tiny little fruiting bodies. And then he'll mark it as positive, and we will take it back to the molecular lab to qPCR some of those samples. And then over here, we have a bunch of our carrots. You can see that we have been doing a lot of carrot baiting lately. So these are for various different projects, all for testing ceratocystis viability. And then we have Josie Tupou over here. Say hi. Hi. And she is actually baiting some samples that we caught the other day in the field. I think we actually have a video of Dan and us doing that. And so what she's doing is taking some carrots that she already cut into little discs and popping them in the little bag with the sawdust. And then she'll spritz it with a tiny bit of water to help the Ceratocystis grow if it's there. And then close it up and we'll check it in a few days.
Okay, thank you for bearing with our amateur videography.
Hi everybody, I'm Ellen.
Okay, so now moving on to the direct transmission. So we wanted to see if the ambrosia beetles could actually carry rod-causing propagules on their bodies. And so this is kind of a newer slide. And so we collected the beetles using 3 different techniques, which you can see here. So one was we collected beetles directly from bolts so wood stem sections that are about 20 inches that we knew had Ceratocystis leucohea in them. And we also collected beetles from sticky cards that were out in the field on ohia trees. And we also had baited traps out in the field as well. And so we used an assay to be able to tell if where we could culture Ceratocystis from the bodies of the beetles. And here you can see we saw that there were this green bar here across all 3 different collection techniques. There is a certain species, Xyloborus affinis, that we think is more likely to be carrying Ceratocystis propagules on their bodies. And if we combined all of these data, we saw that any beetles that you catch or we tested across the board was about 2.5% of the beetles had Ceratocystis propagules on their bodies. And so that number might sound small, but comparing the number of beetles that we actually tested to how much is out in the environment, and especially because we're dealing with Ceratocystis, a single beetle going to another tree could kill that entire tree. And so how we cultured Ceratocystis from these beetles was what we used a technique that we call our carrot aeration technique. And so they're just these tubes with little carrot sticks in them, julienne cut carrots, and water, and they're hooked up to an aquarium pump. You can see over here. And so the aquarium pump just aerates the water, and we let it go for 2 weeks. And I have like a slow-mo video for you guys. This is basically what it looks like, and we let it go for 2 weeks. And so I wanted to point out this picture here too, so you can see sort of, there's actually a beetle right here, and from there you can see all the Ceratocystis is growing out around it. All right, and then we wanted to be able to prove that the beetles could directly transfer Ceratocystis into ohia seedlings. And so we did what we call challenge experiments. And we did this by letting the beetles crawl on Ceratocystis cultures and then put them in these special chambers, which are really just gelatin caps attached to the ohia seedling. And we forced the beetles to attack the seedlings. And then if there was any wilting, here you can see, this is a dissected ohia seedling, and you can see this staining caused from Ceratocystis leucohea. And so we then re-isolated Ceratocystis, recultured it from the seedlings, and we're able to see both Leuco and Huliohea were transferred by Saxonii, affinis, and ferruginous. And so we only tested these guys, not all 5, based on the results from the previous slide. So now that we know that ambrosia beetles can spread rod both directly and indirectly, so what are we working on studying now? So we've really moved towards a more kind of like management approach. And so this is I guess a fun one that actually came up by accident. So this project was started by Lisa Keith, and who's talking after me. And so we wanted to look at, I'm sure you guys are all familiar with this too, the disinfection of different Ceratocystis propagules. And so I'd say probably back in like 2015 or so, Lisa found that Lysol, Clorox, isopropyl, and ethanol were all great disinfectants for Ceratocystis, leukohea, and huliohea in culture. And as we learned more things about how rapidohea death could be spread, the community actually started asking, oh, what about the frass? Like, how can we decontaminate the frass? And I want to say that maybe even Amber asked me about this. And we said, hey, yeah, we can test the frass. And so we actually got a pipes intern to start some of this work. And so we found that actually 5%, 10%, and 20% bleach weren't effective disinfectants of Ceratocystis propagules in frass. And we were able to see that 50% isopropyl, 70% and 90%, as well as 50, 70, and 90% ethanol were all effective disinfectants. And then we took it one more step further to make it more realistic to what would be happening in the field. And we soaked the frass in for only 30 seconds. So it'd be kind of like the same thing as just spraying something, like spraying your boot. And we were able to see that at every single instance that we tried, it worked. And so now I'm going to show you guys what a day in the field is like with us. And so this is a project where we have artificially inoculated trees, and we're tracking how ambrosia beetles, like the assemblage of ambrosia beetles that came in, and how long they can live in the trees and kick out viable propagules in their frass into the environment.
All right, here we are at another field site we have off of Stainback Highway near Hilo. This project is our inoculation trees. So these trees we artificially inoculated with Ceratocystis leukohea either a year or a year and a half ago, and we've been closely monitoring them every week, or as of more recently bi-weekly. During each collection we have sticky cards, which Ellen is collecting from right now, which are to collect ambient beetles so we can decipher the peak times that beetles are attracted to dead and dying trees, and also when those numbers start to decline. We also have been closely monitoring the number of galleries on the tree, so we mark each new one as the tree starts to die and beetles start to attack it more and more. Even more closely monitored are our BDTs, which are the boring dust traps. So we'll take a subset of galleries and put on special boring dust traps that will collect the frass from the beetles, and then we're able to collect that and sample it at the lab by carrot baiting, so we can know, and we'll do that every single collection so we can have a very fine-grained understanding of when viable inoculum is coming out within the frass, and kind of get an idea of when it may start to decline. So that is our inoculation trees.
Okay, so this study is still ongoing, but we have found that the trees can produce viable inoculum in the form of frass for as long as 500 days. We have also been looking into different tarping and insecticide research, seeing if tarping can reduce beetle numbers and also reduce the viability in the trees themselves. I can tell you that tarping does help, but there's also this, cost thing that you have to think about as well. And so we're still working on writing that stuff up. Some newer stuff that we're working on is looking into using different repellents. And so repellents can be used to really manipulate. And so we refer to them as semiochemicals. So you can use different semiochemicals that will, attract or repel ambrosia beetles. And we've tested some products that are already available, such as, so we've tested Verbenone and also Beetle Guard, and they do both look effective. And so we're working on, I guess, continuing the study. Like, we've only done a pilot study so far, and so we're going to be expanding them. And this is a little teaser into my PhD work. So I'm looking into the volatile organic compounds that are produced in the entire Rapidohia death complex. So the compounds that are produced by trees, that are produced by the fungi, and if the beetles are attracted to different compounds. And then we can use those compounds in order to help with early detection. Knowing what the compounds are could help with, sniffer dogs, handheld e-noses, if anyone can get them to work, really. And also to develop specially formulated semiochemicals where you could kind of do like a push-pull system with these beetles. And so these pictures here, this is actually the way that I am collecting the volatile compounds from fungal cultures. So you can see this here, and I will also be doing some bioassays as well with the beetles. So at that, I want to thank everybody. Thank you for letting me be here and talking, and thank you for coming to the talk. And really want to thank JB Friday too, because all those videos, well, everything is all lab space that JB is letting us use at CTAR. So none of it would be possible without JB, actually. And really all of the work that we do is part of, a really big group that I'm sure you guys are aware of, but, shout out to BISC, like they've brought back all the bolts for us and everything. And so with that, I will take questions. That's my email if you wanted to shoot me an email, and that's also my Twitter handle if you want to follow me on Twitter.
Great, thanks, Kyle. I just want to say, wow, just amazing job you guys do, and it amazes me every time how you guys can find these galleries with your eyes. They're so tiny. But I just want to make a mention that what Kyle was talking about with the beetle study, as well as the decontamination and decontamination alcohol study, those are published works that Kyle had written up. And so if you guys are interested in reading further, that is on our website, our Rapidohia Death website. So Kyle's been very busy writing everything. So, our whole team has many different people, but Kyle's been extra busy writing. So thank you, Kyle, for all of that. I do have a question from Jeff. He asked, is it known if ambrosia beetles actively harvest and move fungi propagules, or is it just happenstance? And second part of the question is, can they digest or process wood tissues without fungi, or is it necessary for their life cycle?
Okay, what was the first, or is it just happenstance? Oh, okay. Yeah. So ambrosia beetles do harvest and move their fungi. So they have like little pockets that they store it in, and then they go plant it someplace else. And so what's happening here, what we're pretty sure, but we haven't scientifically tested it yet, but I will soon, is that they're not actually moving the Ceratocystis propagules in their pockets. They're just might be moving it on accident, be stuck on their body. So they're not eating the Ceratocystis fungi. They have a different fungi. So in some systems, the ambrosia fungi actually can be pathogenic, but we're lucky in the case that it's not. And what is the second? And so some of the species, most of them don't eat the wood tissues, but some of them will. So Saxosenii will. What is it? Or is it necessary for the life cycle? Yeah, so they don't, I mean, there's no like nutrition coming from the wood itself. But yeah, one of the species does eat the wood tissues. So Curtis Ewing was a believer that the native also maybe ate some of the wood tissue. Yeah, so I don't know. That's my answer. Sorry.
No, thank you. And I think I have one more question. Why do you feel that one species of beetle, Aphanis, is more likely to carry fungal propagals? Is it something about its biology?
Yeah, so I'm thinking that it's probably they are a little hairier than the other ones. And so these guys are actually pretty hairy if you'd ever looked at them under the scope. And Aphanis seems to be hairier than the other ones. So I think it would be more likely to get stuck on their bodies or maybe something with their behavior too. Maybe they don't clean themselves as much as well.
That's good to know. I guess with that, is there anyone else that has any questions?